Hello and welcome to Programmatic Access to UniProt using Python. For myself, my name's Emily. I'm a biocurator at UniProt, so I put all the biological data into the database. And Aurelian, who works on all the coding and the website and everything kind of behind the scenes at UniProt with me. So today, I'll give you an overview of what UniProt is, and then I'll go through what we're aiming to walk through with you. So UniProt's mission, for those that aren't familiar with the resource, is that we support research, biomedical research, by providing a rich source of freely publicly available information that's stable, comprehensive, and accurately annotated based on protein sequence and functional information. We are a repository for stable identifiers and primary amino acid sequences of proteins, including isoforms and disease variants. We curate physiological function, location, the pathways and reactions that proteins are involved in, their interactions with other proteins and how they're involved in disease. We also curate and range information based on their protein structure and topology. And we have data analysis tools such as BLAST and sequence alignment, which we'll be touching on later. We have two sides to the database, reviewed and unreviewed. At the moment, we've got over 500,000 reviewed entries, and reviewed means they've had publications and peer-reviewed papers added by curators like myself to those entries, whereas unreviewed means that they have been automatically annotated with expertly curated rules, and they're in the unreviewed Tremble side of the database. And we have over 250 million of these. So today's aims are to give you an idea of how to access UniProt data. So I'll give you an example of how you'd see the data on the website, and then Aurelian's going to walk through how you can access that using Python. It'll give you an idea of how to pick the best workflow for your research or what you're looking for. You'll probably familiarize yourself with UniProt and the services it provides, but we do have plenty of webinars available on the EBI training service if you want to go over these yourself. You'll hopefully know where to find help and documentation and how to contact us, either for the website or programmatic access. And you'll be a bit more familiar with how to use Python to retrieve your protein entries, specific fields, filter within your results, and look for certain annotation types and protein characteristics that you might be interested in.
I'm taking over now. Thank you, Emily, for the introduction. And so I'm going to go over the ways to get UniProt data for yourself. So that could be through different ways, and that might be depending on your use case. The first way would be through the FTP. FTP will be making available all the different data file that we have at UniProt, and it would be better for big one-off download and of the whole database all at once. And most of the time, it would require some post-processing afterwards. We also have the API, which we'll be focusing on during this session. This is more for medium-sized download. This is customizable. It works for either one-off downloads of a lot of data, or workflow integration or scripts that you might have to write or do for your own work or research. And you can also download the data through the website. So the website is better for small one-off downloads. It's also customizable, but it's not expected to be able to download the full database through the website. Although we do have a new feature on the website, which is a file generation. So every time you require to download more than 10 million entries, you will be offered the way to generate the file and then come back later to download it whenever it's been generated. It can take a long time, so that's why we will make that available later on to you. But this is still something that you can do through the website. So these are the four main ways to download the data for UniProt data. Yes. So there are multiple endpoints that you can access. Today we're going to focus on the UniProt KB endpoint, which is the main part of the UniProt website and the UniProt data, which is the UniProt knowledge base. But all the sections of the website are also have their corresponding endpoints, and you can see them here. There are also some tools that we provide. So ID mapping, there is a specific endpoint, and also some BLAST and Align that are provided as a EBI service, and peptide search, which is provided by PIR. This is one of the members of the UniProt consortium. And so those, they're not provided by UniProt at EBI, but they're still available for you to use through different documentations. we'll go to a live demo. So I will share a Colab notebook with all of you. For those who are familiar with Jupyter, that's something similar also. And that can be used in Jupyter notebooks, in Python directly, or within Python scripts, or even you can apply what you will see today to other languages. Just to go quickly over the different status code that you might get when you download data. If you get a 200 status code in your response, it means that everything's fine. You can get the data. If there's a 400 something in your response, it might be an error on your side when writing the URL or the queries or parameters. So have a look at the error code that is returned. And 500, that might be an issue on our side. So that could be an issue with the service, and we should be able to monitor it. But if you see that it keeps happening, please get in touch with us. In theory, there should be just temporary issues. And in the response headers, you will see some metadata. So I will go through an example right after, but there are some specific headers that you should look out for. Those are X total results. There is a number of records in the total of the query that you requested. X UniProt release, which will tell you which data release it corresponds to. A link that's in order to paginate to different pages of results, and the component type, which is the type that you've requested the data in. So I will go to the code. So not to worry here too much. I will share the link afterwards, and so you can just follow through here, and I will walk you through the different examples. So here I have a bit of basic setup. So I've stored in variables here some information about the API that we'll be using. So there will be the UniProt website API, which is hosted at rest.uniprot.org. And we'll have some example with the proteins API that is provided by EBI, which is at this URL. And so we'll use that throughout the examples. And here I just have a helper function that will help me query the data and raise if there's any issue. So here I will do a basic search request, and here I will search for everything in the database. So this is what this means, query equals star. This is searching for everything. And then I will pass that as JSON. This is default format. And I will have a look at how many results are in my in the payload written by the server and print it. And then I will also have a look at the headers that are returned in the response, just to show you the example I've told you before. So if I run that, I see number of results 25, which is a bit surprising, especially because I requested everything. But that's because the default search is paginated. So you will get 25 results, but you can see that actually the number of results that correspond to your search is exposed in the X total results. And that corresponds to the big numbers that Emily showed earlier of everything in the UniProt database. You have also the X UniProt release, which shows that this is the data related to 2023-05 release, and other information in there, such as this header here that is useful for the pagination. So this points to the next page in your search. And so in your script, you can query that and iterate through all the different pages by following the links that are returned by the previous page. Okay, let's go back to the slides. So here on the website, if you use the website, you can do some queries. So if, for example, parking and taxonomy ID 9606. So you can have free text search and also advanced search through the Solr format. No need to really know that too much. What I would recommend is to play with these queries in the website. You have an advanced search helper that can help you to create some advanced search. And then you can, through the website, you will have a way to generate the corresponding URL to use for programmatic access. Sorry, just going to show this. So here I have that query, the same query that I did before. So parking and taxonomy ID 9606, which correspond to human. And I will retrieve the data, and it will return some JSON file. I will have a look at the number of results in total, and also print the number of results in that specific page. And so here, let me just run that again. I'm sorry, maybe I should show that first. So if I run that again, I will see in the headers that I have the information of the next page. And here in my logic, I will go through the different pages, and as long as there is a next page available, then I will query the next page. So if I print the number, you will see that in total, it says that there are 143 results in there, but I will go through each page and retrieve this information. So here you see that the total is 143, and I will have paginated through all the result sets. Also, there's another way. So if you don't want to use the paginated endpoint, there's a stream endpoint, which will return everything at once. But there's a bit of a trade-off in there. So it's harder to follow the progress because it will just query everything, but without paginating through things. So if there's any failure at any point, you will need to restart the whole download again. Whereas here in paginated, if there's any failure, you just retry the page where it failed and then continue. And it's also in here it's sorted by score, by annotation score, but here it's not sorted by score. So you will have your all results set, but not necessarily in the same order as you can see in the website. And here you can see that if I do the request, it takes a bit longer than each page there, but in total more or less same time. And then it returns directly the total number. There are other formats that we can request through the API. So here, just to be able to see it better, I restricted the response to one entry, whereas here we have 25 results by default, just to be able to see what is returned a bit easier. So here by default, it will return some JSON. So if I print it just as text, you will see that it's a JSON, so you can pass as JSON. But if I query with format XML, you will see that the return result is in XML. So if that's what you're using in your scripts, that you can request this. There are other formats. The same formats are available on the website, available through the API. So everything that you see on the website, you should be able to do through the API somehow. There's also another way, so through headers. So if you pass the accept header with application XML, you will also get XML. And then there are other formats such as list, which is just a list of accessions that correspond to my search. FASTA. So if you just want the sequences in the FASTA format, you will retrieve all of that here, and you see that they're separated through the comment line here, and you will have all the sequences corresponding to result set, and also TSV. So TSV, which is tab separated values, you will return something that is similar to a spreadsheet, let's say, but this is available here, and so all the columns with the corresponding data is below. And then, so since we have columns here, just as the website, you might have seen that in the website, you can customize your columns. Here, you can do the same thing. So if you specify that you want it in TSV, you can also specify which columns you want returned. So through these fields, there's a lot of fields available. So if I were here, I would go to the website, try it, play with it, and then export the corresponding fields that you want to download, or go through the documentation. You will see that there's a lot of them available. And here I've customized, and you see that ID, accession, length, catalytic activity comment, and this is what is returned here in this specific order. Sorry. There you go.
Thank you very much. So I'm going to go through how you can return a simple entry on the website. So you could do many types of search on the website. So if you go to the home page, you could just find a protein. You can type pretty much anything in there and you'll get a results table. You could type a protein name in. Here we've typed in an accession ID. You could type a species in. You can type a synonym for a protein name or a gene name. There are multiple ways of querying the database. And if you want to put together an advanced search, which will give you a better idea of how to retrieve stuff using code as well, if you click on the advanced link in the search box, it will give you a query builder. So that's worth exploring to get more specific queries back from both the website and using your code. So in this case, we've entered an accession ID. This has taken us to human parking. for PRKN. This is Parkin is actually a synonym. And this is what on the website a protein entry page looks like. At the top, you have all the basic information for the protein. You have its session ID, its name, its gene, whether it's reviewed or not, and what organism it's involved in, and the amino acids. Then you also, at the very top, will have a function comment. So what the endogenous function of this protein is, if there is one. And then you can use the links on the left-hand side of the page to jump to the section of information you would like to view. We also have a feature viewer and a variant viewer, which I'll mention later. There are links to curated and suggested publications and a collection of external links that link into this protein entry as well. So that's how you gain access to a single protein entry on the website. I think if you want, we're going to show it using Python now.
If you want to do the same thing, then programmatically, what you would need to do. So here's just a slide. I'll jump back to the example here. Before, we've used the search endpoint to be able to do a search, but if you know already which accession you want to access, what you can do is retrieve that accession directly. So it's not search anymore, it's directly the accession. And so if I print what will be returned by the API, you see that there's a lot of data. That's all the data that is in the entry page. You will see it here. So one interesting thing that you can do is that if you're interested only in a specific aspect of the page, only a specific bit of data for that entry, you can, similarly as the customization for the TSV before, you can do the same thing in your data, even for the entry. So here I will say I'm only interested in the catalytic activity for this entry. And you can see that if I request that, I will get a smaller payload in there and just the information that I want to see. So you see that in the slide, this information here corresponds to the information in the website that you will see in this way, with the evidence code also and the link to a publication there. And so we can explain to you now what the evidence codes are about.
So within the website page, there are many types of annotation, and you can filter and search these using the API as well. But first, you need to understand them. So as you scroll down a protein entry page, you're going to see these little gray tag boxes. We call them tags as we curate because we like to say we've tagged that piece of information so we know where it's come from. So if you see a tag that says 2 publications, like you can associated with the EC number here, you can click that little gray box and it opens the gray drop-down box that you have, and it will detail the publications that are associated and show data for that EC number or whatever information you're looking at. If they're publications that's been added, they've been added by a curator. Okay. You can also have imported tags. So on the alternative name for Parkin, we have an imported tag. And if you open that, it tells you that's come from HGNC, which is the Human Genome Nomenclature site that we use. Again, we've got another example in subcell localization over here. We've got a curated publication in the nucleus saying this protein is found in the nucleus. And then we've also got a bisimilarity tag for mitochondrial outer membrane. So what we mean by that is if this is the human protein we're looking at, then there's been data found in the mouse that says the mouse protein, Parkin, is found to be in the mitochondrial outer membrane. And because the ortholog is human, we've copied that information by similarity over from mouse to human. So that's what similarity tag means. You're going to find these tags throughout the database and throughout the entries, okay? So if you can have the next slide, these are the evidence codes that you're going to find behind these visual tags. So if you were to look at it in code, these are the ECO codes you're going to find. So 269 is experimental evidence. So that's what a publication code would be. If data's been found in a publication, you're going to add an experimental evidence tag. If you're adding by similarity, because the ortholog has the data, it's going to be 250. 305 is a curator inferred data. So this might be data that a paper doesn't go far enough to say, this definitely does that. But the curator probably knows, and by orthology and other information and being experienced in that field, that protein is involved in a family that is known to have a role in that specific system. In that case, we would have a curator tag. 305. Or we can import evidence from other sources, which is 312. So this will be like an import tag from Expression Atlas as an example, because they have evidence that we've then imported into our database. There are many of these tags. That is not a full list by any means. Please see the link if you want a full explanation of them as well. So this slide shows you how that would then be shown in isoforms, because we don't just show the canonical sequence, the master sequence for a protein in the website anymore. We show it and break it down into isoforms. So if you're in subcellular localization for protein entry, such as we are in this example, the very top comments are going to be for the canonical sequence. So they're the papers that don't show specific expression data in this case. So the antibody will be general. The antibody might bind to all the isoforms. So we put it in the general comment. The information that's specific to isoform 1 and 2 in this case will be from papers that show specifically that antibody only binds to isoform 1 and is only seen in the nucleus or the cytoplasm. And it's been proven not to bind to the other isoforms. So it's a bit more specific. And again, we have the tags. The tags will be specific to that information in that isoform.
And so to retrieve that programmatically, you will have a way to do your search and also include the isoforms in your search, which is through the specific field. So here, if I take this over example, I can see that I will retrieve also in TSV format and with the specific field, so accession, the function, subcellular location, PTM, and sequence. And here, if I run the search, you will see that it's not really nice to read here, but that's it's separated through tabs here. So you will see that the canonical for this entry has this function written here, and the isoform 2 has this other function, which is slightly different. And so you will see that would be the way to separate those annotations from the different isoforms.
So now we're going to talk about variant data. So variant data is displayed all over the UniProt website. Little bit of nomenclature housekeeping. In UniProt, we describe a natural variant as a variant that causes a disease. It occurs naturally in a patient, and as a result, that patient develops a disease. A mutant or a mutation in UniProt is described as an experimental variable. So someone's taken a mouse or a fruit fly or a cell line, and they have made that mutation in that line from an experimental point of view to prove that that residue or that section of protein does X, Y, and Z. So when we're talking about natural variants, we're talking about disease in humans, and it occurs naturally in the population, in the disease population, hopefully as well. So in the protein entry page, you have what you see on the left of the screen here. You have a disease and variant section. And if it's specifically associated with a disease, like Parkin is associated with Parkinson's disease, we're going to import the description from OMIM. So you'll see we've got tags here with publications, and in them publications, we have significant variants that are significantly associated with Parkinson's disease, which means we can then say, yes, involved in Parkinson's, this is a description of Parkinson's, that's from OMIM. Further down that section of the page, you have a variant viewer and a table, and you can use both interchangeably or together. This will show you along the entire sequence of the amino acid sequence for this protein, from 1 to whatever your other terminus is, in dots, or the variants that are found and associated there, and you can filter between them, between consequence, so whether they're disease causing or benign or unknown, and also the source where they're from, so whether they're UniProt, whether they're imported, or whether they're from somewhere else as well. And each of one of these dots is clickable, and when you click them, you get opened the ELA tooltip, which gives you further information. You can also view these variants on the feature viewer, which puts them into the context of the wider domains and features within the protein. Or you can view them in the variant view, which is what we're showing on the right here. And that is where all the variants from all the sources are provided in one large graphical display. And again, you can filter by how what their consequences, whether they cause, whether they're likely pathogenic, if it's unknown, and again the provenance, so where it comes from, whether it's UniProt curated or imported as well. And when you click on one of the variants in that graphical display, it will jump you down the table below there, and it'll highlight in yellow the variant that you've selected and tell you further information on that specific variant. So if it causes the disease, how it might cause that, if they've done functional analysis, the evidence tags associated with it, and its source as well.
And so to do that programmatically, I will jump back to the code example. So taking the example that Emily was showing just before, one thing that you can do is, similarly as before, when you customize the fields that we wanted to return, here I want to return only the field FT variant. So if I have a look at the response for this, you will see that there will be data about that variant, the original sequence, the alternative sequence, and some of the echo codes, the positions, and all of this. So just to show you how many there are in total in this entry, I will just print the number of them in this entry. So here I have 40, and this is coming from the website API, so the UniProt website API. We also have the proteins API that I mentioned earlier, which will include all the data that is already in the UniProt website API, but also add some imported information, in that case from CleanVar or the large case studies, and they will have their origin returned, but you will see that there will be more than 40 in this case. So if I print that, I will see so that the UniProt website has 40, but the proteins API will expose 599. There's a bit more in there. Those are not necessarily being curated by UniProt, but they will include the ones that have been curated through UniProt directly. Okay.
So now we're going to talk about how to retrieve and compare orthologs on the website. So if you want to compare orthologs, I've done a basic search here. If you want to compare them using an alignment in this case. So I've done a basic search using parking, which again is a synonym. But in this case, it returns a set of results, and we've added 4 of them to basket. So this is the first time we've shown you a results table in the webinar, and really almost talked about all these categories that you can filter for. If you want to familiarize yourself with all the data categories we have on a results table, any results table, click Customize Columns. Customized columns will give you all the options for all the sections in a protein entry page. And you can add and take away what columns interest you. And when you click Save, it will produce a results table like that, but it will include all of your selected information, and then you can download it. So the example here, I've just got protein names, organism, and amino acid sequence length added just because that's how my basic one is set up. I've got the parking search results, and I've added 4 of them to basket. So I've done that by clicking the tick box on the far left, and then I've gone add next to the basket icon. And those 4 are Drosophila human, rat, and mouse, and I've added them because they're quite similar to one another. Next slide, please. So when you've added them to basket, basket's a really cool function. Basket's in your top right-hand side of the web page. Familiarize yourself with your basket. You can do a lot of things with basket. You can just save stuff for later if you want, but you can do blast searches within your basket for the entries that you've got. You can align all of them or just a few of them. You can download the data from them specific entries and view them in different formats as well. So to compare and align those 4 sequences I've got in my basket, I've selected all 4 of them and I've clicked Align. That opens a Run Alignment window, which I haven't shown here, but it essentially auto adds your FASTA sequences in there. And I've named the job PRKN_align. And the reason I've named it something is because it will keep them results for a week, so you can always come back to them. When you hit go, it then moves to your tool results. Tool results will start with a status saying queued, then running, and then when it's completed, the completed text will turn blue, and you can click that, and that's the link that takes you to your results, which is shown at the bottom here. So this is based on sequence similarity, and within this section, you can customize it as well. But the basic information here is that the darker the purple, the more similar your sequences are between one another. The lighter purple shades are that, you know, there's one sequence there that's not matching. The dashes are where you're missing amino acids. So you can see here that the top sequence is longer at the initial terminus than the other three. And if you probably can't see on this slide, but when you have a go yourself, you can select and highlight different properties on them sequences. So if you want to go find an active site or something along them lines and compare them amongst your orthologs, you can do. These tools are really useful to just go and have a play with them and see what data you can get out of them. So now we're going to show you how to do this using Python.
So the same flow that Emily described, you can do programmatically. So let's go through this code example. So here I have the 4 accessions that Emily had selected manually in the website before. So here I have them listed. That could be coming from a search, previous search, or a file that you have locally. And so you can list those accessions. And then I will retrieve all the sequences corresponding to those. So I will use this accessions endpoint and list a list of accessions that I have here. So here I've just joined them with a comma. So in the end, it will be accessions equal 060-260, comma, Q7, etc. And if I'm interested in aligning those, I will need the FASTA. I will need the sequence information in order to be aligning this. So here I can show you what this query gives me. So this query will give me the FASTA of all the entries that I've listed beforehand. From then, where you can go is then to the cluster omega service, which is one of the services provided through by the EBI. And so here you will be able to do a request for an alignment. So you have to pass some information, so an email, some parameters of the alignment, the output format that you want, the order of the output, and the input sequence. So you have the access to all the documentation here. So whenever I send the link later, you can follow that and have a look at all the parameters that you can customize here. So I will send that request to the service here, and then I will immediately have a look at the response. So response will give me the job ID, which is the exact same thing that is displayed on the website in your dashboard. And then I can have a look at the status. So I can query the status of that job and see if it's still running or if it's done. I will just type this and run it so you can see all of this. So I retrieve first the FASTA, then I post the FASTA to the service. And so I get a job ID as a response, and I need to keep that to be able to request for more information afterwards. So you can see that right away, if I request the status, it will say running. And cluster omega is usually fairly quick. So now by the time I've taken to speak, if I do this query again, it should be finished. Oh, it's still running. Okay. And again, running. It depends a bit on the usage of the service. So in any case, whenever you have that is finished, whenever that is completed, you can then retrieve the job information through this URL, which is also documented up there. And so if I do that, I will get the response, which is this alignment in the cluster omega format, which is what you're seeing here. You can see similar view as the website, but just the difference is that here you have the standard output of the tool. Even if you were to run that locally on your machine, you install cluster omega on your machine and run it, that would be the same kind of output that you would get. The difference with the website is that the website will enrich the data. So you will have links to be able to jump to that entry, or you will have the annotations that Emily described before. And so if you have domain information, or if you compare different annotation across the different orthologs, you can do that through a website. And to be able to do that through programmatically, then you will need to combine this output with the information that you can retrieve programmatically, as I've showed before, the catalytic activity or these kind of things, or the domain information and all of that. But programmatically, then you will have to do that yourself. Let me just go. Okay, let's go to this.
So now I'm going to run you through the Retrieve ID mapping service. So this is on the website. You can access this from the home page or on the top toolbar along the top of the website, as with all our tools. This mapping service allows you to convert lists of identifiers between different databases. So the example here I've chosen is UniProt accession IDs to RefSec nucleotide IDs. That's quite a simple conversion. You can use this for stuff like changing gene names to different IDs, ensemble sequences or transcript IDs to gene names. If you go onto this service and click the drop-down box under the from database to database, there's loads of different identifiers, and you can even type and search within the boxes as well. This service is really useful if you've done something like mass spec analysis and the software that you've used has spat out a set of identifiers that the next piece of software, maybe doing some Go annotation or something along them lines, doesn't understand. So you can put them IDs into UniProt and convert them to other IDs that you can use in your further analysis. And you can copy and paste your IDs into the mapping service, or you can upload them from a text file as well if you've got a lot of them. So you just put your IDs in the box, or you upload the file. You tell it what the input is, what sort of IDs they are, and what you want out of that. And then you hit map, and it will come back with a set of mapping results, and it will give you a simple table of from and to. It might even, depending on how you've got UniProt set up, it might even give you some protein names and IDs. So when we show you results tables, that tends to be specific to the person and their web browser that they're using, because I like to see that when I search protein. Yours might look differently because you like to see the function comment the whole time. If you click customize, just like I mentioned in the previous slide, you can add and take away whatever information you want there. If you want to download a simple text file that just includes the identifiers that are your output, because obviously you've already got the input, then all you have to do is you get rid of all the columns in customize. Click OK, and you'll get a box like this on the bottom right, and then you just click download in whatever format suits you. So that's really simple. You put one set of IDs in, you tell it it's that type of ID, and I want it converted to this type of ID, and you get rid of all your information in your results table and you download it, and therefore you then have your identifiers to move forward with your analysis. You can obviously download other information from UniProt associated with them identifiers if you want the function comments, if you want the sequences, you want the pathways they're involved in, or any associated Go annotations. You can have all that if you want, but what I'm saying is you can do simple or you can do complicated using Retrieve ID mapping. So now we're going to show you how to do it simply using Python.
One thing to keep in mind with that service is also it's the service provided by UniProt. So obviously there's a lot of in and to databases that you can use, but you always have to go through UniProt at some point. So you can map from non-UniProt to UniProt or from UniProt to non-UniProt, but you will not be able to go from non-UniProt to non-UniProt. So for example, I cannot map directly a PDB ID to a Campbell ID, for example. I would need to go through UniProt at some point. Okay, so let's have a look at that in Python. So here I have my same list of accessions as before. So in the example that Emily was showing, we're mapping from UniProt KB to RefSec. So this would be the way to do that. So I have the ID mapping endpoint, which is similar to a line you would need to post this information to the service and pass the data in there. So in this case, instead of passing the sequences, I will need to pass the accessions because that's the input here, the accessions. And then in the output, I will get a RefSec IDs in there. So this is certainly going to be really quick. So I will now have the time to show you the fact that it's running. So whenever it's finished running, it will redirect you directly to the results. And I believe this is what will happen here. But if you have a lot of accessions in there, if you have 1,000, then you will probably go through that running step. So you will need to do, just as the alignment before, you need to wait for it to have finished before being able to retrieve the results. And you cannot expect to have the results right away. So here, if I run that, you see that I was given this job ID, and when I request for the status, I already got redirected to the results. But yeah, let's jump to that. So if I then just retrieve the ID mapping status with the job ID, I will get, whenever that's finished, I will get the results, and you can see the same thing that you can see on the website, which is the table of from and to, and you will have that here, from and to. And if you have if the two is a UniProt data set, so like UniProt KD or UniRef or UniPark, you will have the same columns. As Emily said, sometimes you might have more columns displayed there that correspond to your customization. And so you can also do that here. You will not just get the ID, you might get more information like the protein name or the gene name or anything that you would have customized in order to retrieve this information. Okay, so yeah, just to let you know, so I will share right after that the link to the collab. You can also scan it through that QR code. Also in the collab, if you continue afterwards, I haven't shown that, but there are some extra workflows. So you have BLAST, you have some retrieving more information like PTMs, you can have some visualization examples. So I encourage you to have a look at that afterwards and play with it and see how that can be helpful to you.
So that's the end of the webinar. I'd like to thank our funders. So we're funded across the NIH as well as from other sources as well. And our team, can I have the next slide, please? And our team is based across the 3 sites in the consortium, PIR in America and SIB in Switzerland. All our staff are based across all 3 sites. So you have curators, developers, and key staff across all 3 sites as well. And there's quite a lot of us now. And this is our help and training page. So wherever you are on the website, there's going to be a little green help kind of tab on the right hand side. There's a little help and contact us button in your top right in the toolbar. There's always a little feedback tab as well. If you're really happy about something or if you're really not, let us know. Or if something's broke, also let us know. On the home page, we have our help and training section, which will tell you where our up and coming training is next, where our past webinars are, and where you can access the training at all 3 consortium institutes. And as part of EBL, we have some upcoming UniProt webinars in the next few months. So our next one is not in November anymore, is it, Anna? We had this conversation. It's on the 14th of December, not the 30th of November. But that's with Michelle. That's on covering protein function. So that's how we curate protein function into the database. On the 18th of January, we have a webinar with Pedro, who's exploring automatic annotation. On the 8th of February, we have sequence analysis tools. And then there's another webinar with me on the 29th of February that explores UniProt and how you might use that if you're looking at proteomics data. And we've also got some, I'll let you do your bit.
Yeah. And regarding the website, so as I said at the beginning, we have the file generation, which is fairly new. So I encourage you to have a look at that, try the feature and download. So that would be for download of big files, but through the website. There's also a new genomic coordinates tab in UniProt KB entries. So you can have a look at that and try it out and give us feedback. This is fairly new. So this is, we're really welcoming all the feedback on that. And the next data release will be in January 2024. And so here are the list, here a slide about the EBI webinars. So you can see, maybe Anna, you want to cover this? Or?
Yeah. So we have another series running at the moment on Wednesdays, which is the molecular building blocks of life. And that's covering a lot of EBI resources. And next week's You can see there is on transcriptome, single-cell transcriptome data, but we'll be continuing that series for a few more weeks. And then, as Emily mentioned, the next UniProt webinar on the 14th of December. I've just dropped the link for the upcoming webinars into the chat, so you'll be able to find both of those available for registration now.
Okay, and now we'll open the floor for questions. So we received some questions already in the Q&A tool, so we can go through them.
Yeah, so there's one I want to answer first because it was my mistake, basically. So someone's asked, when we were looking at the ID mapping, that I said, you can customize your results table. Now, obviously, in the example I did, we were going from UniProt IDs to RefSec nucleotide IDs, and it turns out there's no customize option. And you're right, there isn't. If you do it the other way and you're going to UniProt accession IDs, you can customize your results table. So presumably your customize option will be if you're going into UniProt IDs rather than away from them. So yeah, I'll correct myself on that. Sorry. I obviously don't go the other way that much. So I'm gonna go back up here. I have a spreadsheet with a few hundred of UniProt accession numbers of proteins of interest. Is there a way to query all these accession numbers without using the loop, I'm assuming?
No, for loop. That would be a for loop,
For loop.
in Python. So
not
yeah,
programmer. I apologize.
it's fine. Yeah, I think that so this question was asked before we went through the ID mapping slides. So obviously you can, if you have a list of accession, you can do a for loop and just query each accession one by one. But yes, if you have a few hundred of them and if they are coming from a file, what I would recommend is to use the ID mapping tool. So in the same way I've shown in the code, so I have a list of accessions. So in that case, that list would be coming from your file. You would need to read that file and generate a list, and then query the send that list to the ID mapping service. And you would specify as an input the UniProt KB accession, and as an output, whatever. So is there a way to query the accession? Oh yeah, so as an output, that would be the same. That would be UniProt KB. So you get from the accession to the UniProt KB data. And this is when you get the customized column that Emily was just talking about. So if you go from UniProt KB accessions to UniProt KB data, you will get access to all the data that correspond to these accessions. And I think there was just the follow-up question by the same person of what is the maximum possible number of accession in the joined, oh, it's just jumped, sorry, joined list in your example. So I don't remember that exactly, but you can put a lot in there. This is documented in the ID mapping help page. So I would recommend to go through the help page, search for ID mapping, and you will have the documentation on how to use the endpoint, how you can customize it, and the different limitations. You have a limitation of input. And in that case, if you have more than that, you can just do 2 jobs. So you can do as many jobs as you need. Basically, you would batch them into groups that would be able to be accepted by the service, and then you will get different result sets. Okay.
So Raphael would like to know, is it possible to customize alignments with different substitution matrices, gap opening, gap extension scores?
Yes. So for that, you would need to customize that through, so following that documentation here, yes, you will have access to everything that is that you can customize in the Cluster Omega tool. So usually that corresponds to, if you have installed that locally and run that locally, you will have a set of options. So I think in theory you should have the same set of options here through the service. And so following this link, you will be able to see everything that is customizable for Cluster Omega for the alignment, and that would include those gap extension scores, for example.
So Valerie, sorry, are you done?
Well, maybe I can ask you a question then.
I don't know about that. Go on then.
Now, just a small, quick question about what is isoform. So maybe we can quickly cover that. Do you know?
Pure biology, isoforms. So we term our canonical sequence the master sequence. So we take this from ENA. And ensemble labels a canonical sequence as well. So we try and keep this consistent across other resources. So our canonical sequence, although it's been generally considered the longest, we're now looking at other data to suggest it's the one most widely expressed. Isoforms are where if you've got, I can't think of an example now, but if you've got a gene and it's got 8 exons, let's say, and the canonical protein contains all the transcripts of them 8 exons. So that's isoform 1, let's say, the canonical. Isoform 2 might only have half of them exons. It might only have the first 4. Isoform 3 might have the last 4. And isoform 4 might have every other one. So they're proteins that share the sequence from the canonical, and they shared the same gene, but they're transcribed differently. So that's what an isoform is. And we obviously compile them in UniProt. So although a protein entry page is attached to the canonical, when you go to the sequences section in the protein entry page, we detail all the isoform sequences as well. And within each section of a protein entry page, if we've got isoform specific data, we do include that. So this probably leads into how can you download or review fast sequences for a given entry, right?
Yes. Okay. So
I'll leave to you.
yeah. So I'll just read that question. There's multiple questions, so I'm trying to follow this. So how can you download via API all the reviewed FASTA sequences for a given entry? Okay. And how can you get the annotations? So PFAM, KEGG, etc. from a list of UniProt IDs. Okay. So that's 2 different things. So to download all the sequences for a given entry, so that would be including the canonical and the isoforms, you will have that information in the entry data. So one of the fields in there, one of the part of the entry data will be the sequence that would correspond to the canonical sequence, and then you have some other information which are alternative product, I think it's called, and so that might be related. That will be the list of isoforms. So I don't have this off the top of my head at the moment, but this will be included if you download one entry, the full payload of that entry. You will also get in there the isoforms, and there would be specified as isoforms. You don't need to specify anything in there. If you want that as FASTA, you could also do a query, and as I've shown before, do the include isoform, a query for that accession. So accession the accession name, include isoform, and ask for the format FASTA. Then you will get a FASTA file with all the different sequences in there. And then the second part, how can you get all the annotations, PFAM, KEGG, from a list of UniProt IDs? So same thing as before, from a list of UniProt IDs, if you want to get information about those IDs, you would use the ID mapping service to UniProt KB, and then you would be able to customize. So in that case, those are external references. And so if you try it on the website and customize the column and look for PFAM or KEGG, you will be able to add those columns. And then in the download panel, you will be able to see, generate a URL for that query. And you will see the name of those, of the fields that you would require for this. So I don't remember also, I mean, there's a lot of different fields that you can customize that are listed in the documentation. But I think PFAM, there might be something like xref_PFAM. Yeah, I would confirm that through the documentation or by using the website. The website will tell you if you play with it and render something on your page that looks like the data you would want programmatically, then you can export this, and you have a useful tool that will export that, and you can copy that URL for API usage.
Just a point, Anna, can we have mine and Paul's webinar from 2 weeks ago copied into the chat, please? Because this webinar is an introduction to the website. So it includes how we format everything, how we customize the columns, how you download the data. So if you're not familiar with UniProt, we've just kind of dived in there and given you all this information, and you're going, what? Watch that webinar, and that will give you a really good start. The more familiar you are with how we add the data to certain sections within a protein entry, it's going to really help you understand what you need to download when you use Python to get the information you need. Okay, so explore that. It's a good webinar. It's quite general, so it's not too specific. And I say anything you do on the website, you can get programmatically. It's just about knowing what to download, knowing what to filter by.
And I'll just share the link to the webinar. So in the chat, if you want to have a look at that.
Thank you.
Okay. Which one should we take?
Well, they're all your questions, so.
Okay, I'll start with the top rated there. So is there a quick way to download a file with name sequence for a rather small list of genes in a way that we input them? For example, all G proteins. I'm assuming that is a category of protein.
The family of proteins.
Okay.
Big one.
It's a big one, so maybe that's not, yeah. So in any case, what you would do is look for your proteins. So I'm guessing this is some kind of name, so you can look for protein name or gene name and look for that. You would get some kind of result set, and then for that you can similarly. So it's just everything, but you see that this will be able to generate for you a URL. So that's not your example because I don't really know how to formulate that proper query, but you wouldn't need to do the advanced query in there. And then you try to download it. And then if you can either download it or retrieve the URL that you would be using in your script. So if it's a small list of genes, you could just download that through the website, I guess. You scroll there, you make sure that you have all the accessions that you want, then you download. If it's a bigger list of genes, then you might want to do that programmatically.
So the category Katrina is specifically looking for in this question, because this isn't immediately obvious to anyone that doesn't curate. The category you're specifically looking for is CC similarity. So if you go onto an advanced search, and instead of gene name, you type similarity, you have family and domain / sequence similarity. Yep, and then you would type G protein, hit go, there you go. And then you can filter by species and by anything, by reviewed, unreviewed, taxonomy, etc. So if you're looking for a specific family of proteins, as we curate the entries, we go, hey, this is G protein, we add it to the G protein family. So there you go. I'm not saying it's all encompassing and inclusive, because there's obviously got to be data out there that says that protein's involved in that family in them pathways, but yes, that's the best approach to use that information to download them.
And then you will have here the query that you can use in your scripts to be able to do that if you don't want to do that only through the website. Okay. Is there a way to increase the number of entries from 25 to a significantly higher number? So if you using the search endpoint, which is the one that is paginated, you will have by default 25. You can customize to any number, and sorry, up until that limit. And so that limit is 500. And so you would still be able to download pages of 500. But yeah, if you want more, either you have to go through the script, just like I did before. I've shown you in the examples higher in there, or you will need to use a stream endpoint, which is the other endpoint I've shown also in the collab. So stream endpoint, again, this is just one query that might take a longer time, but you will retrieve everything at once. And the paginated endpoint, it's multiple queries that you have to go download one after the other, one page after the other. And if there's an issue during the download, you can resume from the page you were at when it broke. Okay. Is there any limit in UniProt for the extraction of FASTA sequence and EC number using Python code? Not necessarily. That would be the page limit, as I said before, or that could be if you do a lot of queries, you might not be able to query all of the UniProt entries at once. So you would need to, there would be a rate limit. You're not able to do millions of queries in the same second. But yeah, apart from that, there's no specific limit. You would need to customize your query to retrieve the information that you want. So the sequence and the EC number, which would be specific fields that you query. And that's about it. Okay. Is it possible? So I'm going to skip over the, do I need to install Flask? I think it's, no, yeah, this is not, we don't need to install Flask. You can just use Python right away. I think Flask is on top of Python. You don't need to do that. You can just use the examples I've shared right away in Python. Is it possible to filter by maximum protein length? So yes, that would be the same thing through the advanced search. So let's say advanced search. On top of that specific query, I want to add length. And I say, okay, from 0 to, I don't know, 1,000. And then I will get only the entries for which the sequence is less than 1,000. So I think here it's the same. Let's reduce this a bit. There's a bit less, I think. Yeah, but you can play with that and you can put any value, and then you will be able to filter this way. And again, after that, if you want to use that programmatically, you will get that query in here. So here the length is specified in this section. Okay. 2 more questions. In the question you just answered, okay, I'm not sure when that was. I'm not sure the part in a way that we input them. For example, in the order that we input them, I wasn't able to get the entries in the same order as on the website. How can I get this? So if you want the entries in the same order as the website, you would need to use the search endpoint, which is the paginated one. So there would be, I mean, if you've sorted them by a specific order, you can retrieve them by a specific order, and then you go and download them. They should be the exact same order as you see on the website. If you use the stream endpoint, which is the one that returns everything at once, they might not be written in the same order. This is not guaranteed. This is for performance reasons. You will then have to, if they're sorted in a specific way, so for example, by sequence length, you will need to do that programmatically afterwards and say, okay, in my result set, I will sort them by sequence length, for example. Or if you want to sort them by a specific thing, you will need to query that specific thing so that you can sort it by that. So for example, the annotation score, you could retrieve that specific column and then sort by that. That would be the way to download in the same order. Oh, sorry, in a specific order. And then finally, in the website, I can see the publications are assigned a category function sequence. Can we filter that in the API? For a list of UniProt IDs, see only publications related to function. I'm not entirely sure. Emily, do you know if that's possible?
I was hoping you'd know. I think this would be a really cool way of getting data out of the UniProt. I'm just not entirely sure how to do that. I know within let me have a look. Within an entry, you can search publications and you know what tags are associated with them.
Mm-hmm.
I don't think it necessarily tells you.
But that programmatically, then if you want to search for specific publications, then you might, you can do that through the advanced search. You will, I guess, either search by a specific publication, by PubMed ID, or by types of publication. I'm not entirely sure what you would filter on. But
well,
you might
must have some way of doing it, because just look at if you go to go to any UniProt protein page, preferably human, something interesting. I say this like you know what interesting looks like, right? Hit publications, and then if you scroll down below the sequences ones, so I mean,
sorry, the entry.
No. Back to publications. So here you go. It says this publication is in category PTM processing sequences, like in the main body of the body of the publications. So we do tag them because these are reviewed, so that'll be tagged in them. And like the second one is a sequence, so it provides a sequence to that entry. So we must have some. We must have something somewhere.
Mm-hmm.
Now, how you can access that to filter it, I don't know.
That's the publications endpoint. So I would say this is also documented in the help somewhere. So someone shared the link to that. So that works if you want to filter within a single entry for specific publications. I thought it was the other way around of if you want to look for entries with a specific publication on that section, I don't think that's doable, or you would need to filter that afterwards. You would get all the entries with that publication and then filter if they correspond to that. But yeah, for a single entry, that would be the way.